Good morning. I'd like to thank the NASPS board for the opportunity to present our research here today, and to you all for participating in this event. I'll be talking about recruitment bottlenecks today during early life stages of lake sturgeon from a 19-year perspective based on our research program on the Black River population in Michigan. I'd like to recognize my co-PI, Dr. Ed Baker, for his longstanding collaborations, and I'd like to recognize the many groups who have provided continuous funding support during this 19-year period. Also, a huge shout out to all the graduate students, technicians, past and present, who have done the heavy lifting to collect and analyze the field data to make the project a success. Just as a bit of an outline here, I'll start out with an introduction to our system, the study area sampling methods, and a bit about lake sturgeon ecology. I'll spend most of the time talking about the stage-specific biology, describing mortality levels and identifying causes by stage and cumulatively across all stages from egg deposition to age 0 fall fingerlings. And then I'll provide some conclusions and suggestions for management. The study area is the Upper Black River in Michigan. It's a suitable study for a number of reasons. It's relatively short. It's about 11.5 kilometers of free-flowing river. It's about 25 meters in depth with meters in width, with an average depth of 1 to 2 meters. It's a moderate population size. We estimate based on a novel closed population mark recapture estimator developed by Shirley Plager, that about 1,200 adults. It's closed to immigration. All spawning locations are known. We've tagged almost the entire population. Over 18 years, data are available on 4,354 captures of almost 1,400 individuals. And most of the adults have been captured multiple times. On the right is a picture of the river from the confluence with Black Lake up to the spawning areas. You can see the gear that were deployed to collect data. We have passive pit tag arrays at the mouth immediately below the spawning areas and throughout the spawning areas. We have temperature flow loggers throughout the area, and you can see there's a 3-kilometer area that we do visual surveys for age 0 fall fingerlings. And this is just to show some of the gear deployed in the river to monitor adult movements and entry into the river. So lake sturgeon are wildly fecund. Millions of eggs are deposited every year into the Black River. We have about the same spawning number every single year, but they exhibit a classic type 3 mortality curve where there's tremendous loss over the first several months of life due to a variety of factors. So most of the work that we've been engaged in over 19 years has been to describe the biology and to look at stage-specific causes of mortality and lack of recruitment. What we found, and what I'll describe today, is that it's very complicated. It varies based on stage. And there are a lot of players that are predators, that are alternate prey. The effects of physical and biotic features are extremely variable even within years. So the adults enter the river every year early in the morning and late in the afternoon or evening. The graph on the right shows a typical year where you have spawning date and the number of males and females entering the river, as well as the temperature and discharge by day during this period. Spawning in the Upper Black River is multimodal. There are individuals which spawn earlier in the season and later in the season that we call early and late spawners respectively, but over 19 years, adult spawning has occurred over as short as an 18-day period and as long as a 45-day period. Interestingly, spawning by early and late spawning individuals varies in terms of where they're actually spawning. Spawning individuals will spawn typically in areas upstream. Those spawning later in the year, typically locations downstream. As I mentioned, the chronology of spawning varies greatly across years as well as the duration. So here's data for 2 years, numbers of males, numbers of females. I've extended the graph out here so it's concordant. And you can see here that this year, 2021, there were a number of individuals that spawned extremely early. And this is really a bad idea, spawning in early, and mid-April is very hazardous because the temperature regimes during the month of April can be really, really very tricky, as was the case this year. So we had some 25 or so individuals that did spawn. We actually collected gametes from males and females during this time, but we have no evidence that any of the eggs survived. So female to egg. So we've seen that the magnitude of interannual variation in reproductive success of females really is tied to depensatory effects of spawner abundance. So these animals are broadcast spawners. It's a very inefficient reproductive system, requires that there be a lot of sperm in the water, which means you need a lot of males in the water. And oftentimes we'll see females without an adequate number of males that will just sit in the river for days on end and not spawn until there's a sufficient number of mates. And so this is some data from Carrie Dammerman's dissertation, some work we collaborated with Molly Webb on for 2012 and 2013. The information on female reproductive success was based on genetic determination of larvae to those females during each of those years. And so reproductive success of individual females in both of these years was related to temperature. So later spawning females were more successful. Females that were spawning under higher discharge conditions were more successful. And importantly, mate number, the number of males that were mated and with female and the operational sex ratios associated with those females were really very important. So if eggs are fertilized and are deposited, mortality at the egg stage is very high. So this is some data of tick net sampling in transects across and downstream at one study area. It's some work from Patrick Forsyth's dissertation. And you can see we did this twice at day 1 and day 3. And there's, well, the pictures speak to themselves. There's a tremendous loss over this period. Patrick went on to try to identify sources of mortality. So he built this contraption where he had these rebar containers which had different trays. The trays had different mesh sizes surrounding them, which would preclude different size predators and would also impede flow. And what he found was that across these different treatments, mortality was relatively concordant. And over 91% mortality over a 5-day period, which is about 2/3 of the incubation time, irrespective of the mesh size treatment. Mortality is also very high at the larval stage, and we quantify this using larval keyframe nets where we're able to capture individuals. We monitor flow and depth across the river, so we're able to quantify the total number of individuals relative to our sample. So we use 5 nets. The nets are fished for 5 hours in the same locations every night. And we record data on the discharge of the nets across the river, the sizes of and the numbers of lake sturgeon, as well as larval suckers, other fish larvae, as well as aquatic nets. Inverts. And so, you know, the larvae drift downstream, some are caught in the nets, some are eaten, and we use genetic metabarcoding to identify who's eating who. So the following day, we'll go out and we use electrofishing to capture up to 10 individuals of each of the potentially piscivorous predator species, and we use eDNA metabarcoding to quantify predator diets because you can't recognize what's in the stomach the following day. So each of the fish are collected, and this is just an example of Moraniak's work that he conducted in 2015 and 16. So as I mentioned, the survivorship up to the point that we catch the larvae varies from year to year. Again, if we have the same number of adults that are spawning, plus or minus, then what we have, based on the number of larvae we collect, is some indication of the relative survivorship from the egg stage to the larval drift stage. And we can see here that over a 20-year period, 19-year period, we have over 2 orders of magnitude difference from the lowest to the highest number of larvae collected. It's also variable in terms of the number of individuals of all the other co-distributed prey. So this is a graph showing number of sturgeon larvae, number of sucker larvae, and number of invertebrate larvae captured. So we know, again, from genetic determination of parentage, that those fish that are captured earlier in the year are from early spawners, and those later in the year from later spawners. And there are generally higher numbers of larvae produced by early spawning adults. There are more of them than later spawning adults. We also know that the out-migrating sturgeon larvae are out-migrating with different alternate prey communities. So for instance, the larvae from early spawning sturgeon out-migrate with white sucker larvae. Those that are out-migrating with later in the year are out-migrating with silver red horse suckers. The macroinvertebrate community is quite diverse, and we see smaller numbers and very, very large differences in the families of macroinvertebrates represented during the early and later portion of the drift period. So generally speaking, just to quantify what these fish are facing, these young fish emerge from the rocks and substrate, and they're vulnerable to predation. And just to compare early and late drifting larvae, early drifting larvae, these fish generally are larger in body size because they're incubating at colder temperatures. And this is some data from Jamie Crossman here in the lower right-hand side, just showing the magnitude of differences of larval body size by families of early and later spawning adults. Fish, lake sturgeon that are drifting earlier are more concealed by other prey species. There are more alternate prey and greater diversity of alternate prey than we see later in the year. Those individuals that are in the early drift, generally speaking, are timing the out-migration under a new moon. So they're in comparatively high levels of darkness compared to those individuals which are out-migrating later in the year, and thus they're more visible and they're more vulnerable to predation. This is just some data. Again, this is based on DNA barcoding of predator stomach contents. So an individual has or has not sturgeon DNA in the stomach. And you can see here an example of prey shielding. Lake sturgeon larvae are concealed when they're out migrating with a diversity and abundance of higher prey. So this is based on biomass along the x-axis and the probability of sturgeon consumed in a predator stomach. That probability of a sturgeon consumed increase as the biomass of sturgeon increases, and it decreases as a function of the increased biomass of macroinvertebrates. The biomass of catastomids really was not a factor here. Percent of lunar illumination is really important. So here the probability of lake sturgeon larvae consumed increases with an increase in lunar illumination. So the moon helps predators see. We also found that sturgeon were more likely to be in diets of predators on sand substrate compared to rock substrates. Likewise, we found that in nights with higher discharge, you had fewer predators consuming lake sturgeon. So based on DNA barcoding, the probability of being eaten, yes, no, it is complicated, but it depends on a variety of things that are predictive. Predator species, some fish are more voracious predators of lake sturgeon than others. Generally speaking, perkids and creek chubs are pretty high sturgeon predators. Have higher selectivity for lake sturgeon larvae than other species. The biomass of sturgeon suckers and invertebrates, as well as the proportion of the biomass Nightly is important. Predator body size is important. Substrate, sand versus rock is important. Brightness is important. Based on lunar illumination and cloud cover, discharge. We're using experimental mesocosms, flowing streams, to continue this work. And this is some work of a current PhD student, Joe Reedy. So you can manipulate which predators you're using. The prey communities, sturgeons, suckers, we use Hepanageid mayflies. You can manipulate light levels. You can manipulate body sizes by incubating larvae at different temperatures. And this is just some data that Joe had. So you have higher numbers of mayflies, sturgeon, and suckers consumed in a full moon than a new moon. The probability or proportion of lake sturgeon surviving differs by 40%, whether or not there's a simulated moon or full moon. And there are 20% differences from large cold temperature incubated and small temperature, small size, warm temperature incubated. So body size makes a difference in terms of visibility and mortality, as does lunar illumination. So how can we bring this all together? So I summarized what's going on a stage-specific basis, now putting it all together. Let's start with just 1 year. 2018, there were 93 females, 340 total spawning individuals. We take the body mass of all the individuals, so we know the number of kilograms. Based on the Brook and Binkowski formula, we estimate that that produced probably about 38 million eggs. We know based on information about mortality rates, that mortality rates through an incubation period is probably as high as 97%. We sampled and collected the highest number of larvae there. So we estimate based on discharge and depth that we had a gear efficiency, or we collected about 13% of the area and 13% of the larvae. So we have an idea of number of larvae produced, which gives us an estimate of about 70% mortality through the larval stage. And then we go down and we sample the mouth of the river in the lower 3 kilometers of the river, and we do a capture mark recapture estimate, and our estimate was 50. Okay, 38 million. We have 50 fall fingerlings in the river at the end of the summer. And we estimate from other data that overwinter survival based on Crossman's telemetry work or work by Ed Baker and I with small mesh gill nets is maybe up to 80%. So is that one look in 1 year predictable? And the answer is no. We do have about the same number of females, but the sex ratio varies from year to year simply because the interspawning interval for males and females is different. And so some years it's more male biased than other years. We know that the survivorship at the egg stage is really highly variable, particularly very, very bad survivorship for those years where adults are trying to spawn earlier in the year. And with climate change and variability, we've seen that happen twice in the last 10 years. I mentioned again that we have tremendous differences in survivorship to the time that we collect larvae simply based on the interannual differences in the number of larvae collected each year. But the bottom line is the bottom line. So we do, this capture mark recapture estimate at the end of the year, and over 5 years, it's 52, 0, 0, 32, and 50 are the estimates based on what we collect later in the year in the bottom 3 kilometers of the stream. So based on this, some conclusions for spawning adults, we see, low population sizes of numbers of spawners and the sex ratios based on fertility assurance. The depensatory effects are really very important. There are trade-offs in terms of number of adults and the number of spawning areas. Putting all your eggs into one basket, spawning in a relatively small number of areas is not a good idea. Negative effects of unpredictable environments. Climate change and variability is decreasing the predictability of these early year spawning cues. The eggs, females that spawn in multiple areas lead to higher success and higher survivorship of eggs. We see an increasing prevalence due to things like invasive species. Zebra mussels are also an impediment. We do see that differences in water quality can decrease microbial load and could be beneficial for egg survivorship. The larvae, really high predation rates of piscivorous predators. Can't really do that much about that. Similarly, we see a huge effect of different light levels, cloud cover, lunar illumination. Can't do very much about weather and the lunar cycle. Body sizes are really critical. So the larger the body size, the lower the mortality rates, relatively tenuous based on climate change. The diversity in biomass is something that could be controlled and helped. We can increase the abundance of prey by increasing water quality and maintaining connectivity. So with that, I present our COVID 2020 crew sampling and social distancing over a 6-foot sturgeon in a snowstorm in May, and leave you with this slide showing some background on for our website and other information that you can use to contact us. And I'll take any questions if there are time.